A very good morning everyone. So I will be discussing about the paper titled Human Population Dynamics in Upper Paleolithic Europe Inferred from Fossil Dental Phenotypes. This article was published in 16th August 2024. So the journal was Science Advances and it has an impact factor of 47.73. Talking about the novelty, aim and objectives. This is a first of a kind paper. The first it is a first application of ABC to phenotypic data. ABC is nothing but approximate Bayesian computation. So this study it introduces Pheno-ABC, which is a novel adaptation of ABC for skeletal phenotypes, that is the dental traits. While ABC is widely used in population genetics, this is its first application to morphological data, which has opened new avenues for bioarchaeological research. And the largest dental data set for Upper Paleolithic Europe with 450 specimens, the study surpasses existing genetic and skeletal databases, addressing the scarcity of well-preserved ancient DNA. The challenges prevailing genetic relatives, the findings of this study both supported and contraindicated genetic studies. Example, continuity in the west versus replacement in the east. It emphasizes the need for multi-proxy approaches in paleodemography. The aim and objective of the study the primary goal was to reconstruct population dynamics, that is migrations, bottlenecks and replacement in Europe, which happened from 47,000 to 7,000 years ago using dental morphology. The key questions which were seen in the study was did population persist or get replaced during climatic upheavals like last glacial maximum, and where these distinct demographic trajectories in western versus eastern Europe were distinct, and can dental traits reliably infer genetic relationships. These were the questions which were addressed in this study. The materials and methodology. The sources were fossil dentitions from 87 sites across Europe, which was dated using radiocarbon or archaeological context. Trait selection was carried out using 20 ASUDAs. ASUDAs is Arizona State University Dental Anthropology System. Non-metric traits, example, non-metric traits like cusp for number and room patterns were also seen, and it was chosen for heritability and neutrality. Polarity estimation was also done. That is, the traits were classified as ancestral or derived by correlating modern global frequencies without Africa dispersal routes. The methods carried out. A Pheno-ABC framework was done, that is simulated trait frequencies under 14 demographic models, which includes continuity, replacement and isolation were noted, and coalescence simulations incorporated mutation rates, which is approximately equal to 10 to the power of minus 4 to 10 to the power of minus 2. It was approximated for polygenetic traits. The models were tested, stepwise comparison of models which were grouped into categories based on continuity, discontinuity among the east and west population. It was done to reduce the bias. Power tests were also carried out. Classification error which was assessed, and model distinguishability was also seen. The CE classification error was 4% for the toppest model, and random forest classifier was used. Random forest is something, random forest classifier is where it is used to increase the accuracy. It gives more number of decision trees, and this is done to increase the accuracy. They identified best fit models to majority voting. The model 5, it had 74% of posterior probability. Validation was done. Linear discriminant analysis (LDA) it confirmed simulated data, and it overlapped with observed trait frequencies. Sensitivity analysis using a conservative key tooth approach was used. The N401 was the number of samples, which it yielded consistent results. Strength of the methodology. It handled fragmentary data. It accommodated missing traits or specimens via simulation adjustments, and explicit demographic parameters was one of its strengths. Model include migration rates, bottlenecks and population divergences. And reproducibility. These samples, the code and data, it is available on Zenido. and it can be reproduced for further studies also. These were the key findings of the study. So it was, since it was a retrospective study, so it was divided into middle Pleniglacial, late Pleniglacial and early Holocene, which is late glacial. So bidirectional gene flow between West and Eastern Europe, it supported a pan-Gravitarian cultural network, and late Pleniglacial, that is 28 to 14.7. So it stated that in Western Europe, the population replacement by Eastern group at 28 Ka contraindicated genetic evidence of the continuity. In Eastern Europe, there was more replacement, but isolation and bottlenecks during the LGM was seen. And in the early Holocene period, there was a re-established gene flow post the late glacial period, aligning with the Mediterranean or epigravitan expansions. On the whole, there were two models noted, the best fit model and the runner-up model. So the best fit model was model 5, that is, which was seen in West, which was replaced by the East at 24 Ka, followed by LG and bottlenecks. And runner-up model, two replacement events, 28 Ka and 14.7 Ka, but it was less supported. It had 45% posterior probability. Moving on to discussion. Agreement with posterior work, Gravitarian homogeneity, it matches craniometric and genetic data suggesting a pan-European population. LGM refugia suppose archaeological evidence of population contradictions in southern Europe, in the Balkans, in Iberia and Balkans. The divergence were also noted. The Western Europe replacement, it conflicts with Paleogenome showing continuity. Eastern Europe continuity, it disagreed with the genetic turnover. Interpretation, dental versus genetic signals indicate that dental traits may capture older demographic layers obscured by later genetic turnovers. And there were methodological trade-offs. Pheno-ABC, that is a Bayesian classification, excels where DNA is sparse, but it may miss the fine-scale events because there is admixture. The strengths of this study is there is innovation. This innovation showcased by using Pheno-ABC pioneers phenotypic demographic modeling applicable to other skeletal traits like cranial non-metrics. Data robustness through large sample size, that is, we had 450 samples, which mitigates biases from fragmentary fossils. Interdisciplinary impact, it integrates archaeology, climatology and genetics. Transparency of detailed supplements, it was all tabulated. And the limitations is there are sample biases. Sparse samples, sparse eastern samples, weaken regional interferences. Reliance on published data may overlook unpublished specimens. There were also trait limitations which were seen. Heritability estimates, approximately 30 to 60% that were moderate, and pleiotrophy, example EDR gene may inflate correlations. Dichotomization, losses granularity in trait expression, in dental trait expression. Model simplification was also a very big limitation of this study. That is, west or east divisions, they ignored micro-regional diversity, and they assume neutrality, which may not hold for traits under selection in case of molar sizes. And the mutation rate was uncertain in this study. The approximated rates could skew the coalescent timelines. Conclusion and implications. This study demonstrated teeth as a viable proxy for ancient DNA, crucial for regions sometimes with poor genomic preservation. Challenges like single origin narratives by revealing complexes, localized demographic processes. Future directions, merging of genomic or phenotypic data can be done. Test concordance between dental traits and ancient DNA. Expanding samples to include deciduous state and underrepresented regions can also be done. And refining models to add finer spatial or temporal resolution. Example, sub-refugia during the late glacial period. So the bottom line is, this study is, it used a groundbreaking method with minor limitations in sample representativeness and rate assumptions, but this still stands as a template for future phenotypic demographic studies.